Hello, my name is Chino Eke. I am a rising junior in the class of 2022 studying neuroscience and getting a certificate in global health and policy. This summer, I did a joint internship with the Hahn Lab of Endocrinology and the Cell Lab of Neurology at the University of Tennessee Health Science Center that works in conjunction with the Levaner Hospital for Children. Um, the primary focus of the Han lab was to work with the MC4R pathway and see how the effects of the schizophrenic drug lopoxapine might contribute to the regulation of hormones such as ghrelin, which is responsible for the hunger response, and leptin, which is responsible for the long-term food storage response in adipose tissue within mice models. The overarching and future idealistic intention of said experimentation is to possibly help find ways to better improve the nutritional values for individuals who are socioeconomically disenfranchised, strictly due to the environment that they live in because of the lack of nutritionally beneficial foods. So during this internship, I worked with my fellow Princetonian, Sandra Yang, who's also a member of the class of 2022, in analyzing data sets strictly by going through writing our scripts and compiling figures for experiments done by postdoctoral students at the University of Tennessee Health Science Center. The primary data set that we worked with involved 9 wild-type mice, 12 homozygous knockout mice, and 7 heterozygous knockout mice that were received drug lopoxapine injections and were monitored in the CLAMS data analysis setup, which is short for Comprehensive Lab Animal Monitoring Systems. These systems record everything from VO2, CO2, the number of activity breaks in a given system, the movement of the mice, and also their energy output, which can be calculated by a number of the factors that are recorded. And also involves their food intake, which was quintessential to this experiment. In the middle column, you can see some of the figures that were generated by our scripts. The first being the food intake, given the different phases of the experiment and subcategorized by the different phenotypes of the mice. So there is a differentiation between the heterozygous in blue, the wild type in blue, the heterozygous in orange, and the homozygous in gray. And then later on, we also looked at different factors such as ambulatory movement, so the amount of times the mice move around their cage, given their specific phase or their phenotype expression. And then in addition, we also looked at their energy outputs. All these things with the hope of establishing a definitive response to how lopoxapine affects the mice, and hopefully can be extrapolated into humans with future study. That was the primary focus of our internship this summer, myself and Sandra's. The secondary part was working with the Cell Lab for neurology with phantom limb pain and Drosophila flies. The purpose of using Drosophila flies is their short lifespan, and they're also large quantity, which provides a numerable number of data points, and the ability for quick data collection and turnaround. There was an experiment done by a previous postdoctoral student in the Cell Lab studying the effects of amputations on the limbs or on the Drosophila flies. And this was performed through a microarray analysis in which the data was collected for gene expression on a 1 hour and 5 day after amputation basis. What we looked, Sandra and myself looked at, were primarily the grouping of different genetic homologs between Drosophila and humans, with the intention of establishing a good database for future study between Drosophila flies and humans in specific relation to traumatic experiences. And this project, we did literary analysis and also worked with a system called David, who categorizes microarray expression into different groups based off varying factors. We didn't really do much else because of time constraints with this particular project, and our primary focus again was the MC4R pathway and regulation due to lapoxetine. But I do feel like this experience, given the circumstances of the current pandemic, was very rewarding, and I do appreciate both the efforts of Dr. Joan Hahn and Dr. Jack Sau for all their time and energy that they put into making this internship one that is memorable and also extraordinarily experiential. And I would also like to thank postdoctoral students, Clint Kinsey and Aaron Stephenson, who provided invaluable information that could basically made this internship feasible for both myself and Sandra Yang, my fellow Princetonian. Yeah, so I would also like to thank Princeton, more specifically the Center of Health and Wellbeing and the Global Health Program, for sponsoring this internship and allowing both myself and Sandra to have this opportunity to further our interest in medicine and also contribute a little to the field. All right, thank you for everything, and I hope you have a great day.